Good morning students, myself Komal Walde. Today we are discussed about the topic embryonic development. Before we going on our topic, we know that after the fertilization, the product of fertilization is known as the zygote, and the embryonic development is start immediately after the fertilization, and the development of embryo is start from the cell that is zygote. In the organogenesis or in the embryonic development, the development from the zygote is from the process cleavage is going on. The process cleavage, after the cleavage blastulation, after blastulation there is the implantation, or we can say that the nidation. After nidation there is gastrulation and organogenesis, and lastly the parturition, or we can say that the pregnancy and then parturition. The process of the embryonic development is what start from the zygote, means after the fertilization immediately start the embryonic development, and the first step, or we can say that the first process in the embryonic development, that is the cleavage. So cleavage is formed by the mitosis, means the zygote going on the mitotic divisions, and after the single cell it forms double cell and cleavage go on. Again there is the no growth takes place, means the growth phase is absent, only the division is carried out of the zygote, and this division is known as the cleavage. It forms the daughter cells which is product of the cleavage, and the size of the cleavage is reduces, the metabolic activities or metabolic rate is increases, but the size is decreases. Okay, so in the first stage of the embryonic development, that is the cleavage, and after the cleavage the formation of blastomere takes place. After the cleavage the formation of blastomere takes place. Again in the human the cleavage is holoblastic. Holoblastic means what? The cleavage is directly from the zygote, means the whole zygote gets divided and the cleavage is from. The cleavage is holoblastic, means completely the division of zygote is takes place. First one, the first cleavage and formation of two cell stage. This first cleavage is start after the fertilization, after 30 hours. After the 30 hours of fertilization, first cleavage is carried out, and this is the meridional. The cleavage is maybe longitudinal or meridional, and it is equatorial or horizontal. So the first cleavage is meridional and form the two cell stage in which the formation of blastomere takes place. Okay, continue to the next cleavage, formation of four cell stage. The second cleavage is again longitudinal, but it is right angle to the first one. The second cleavage is the right angle to the first one, and it is occurs after the 30 hours of first cleavage. Okay, then second cleavage, after the second cleavage formation of four cell stage, then after the second cleavage the formation of means there is 8 cell stage, and the blastomere size is decreases. The size of the blastomere is decreases in the third cleavage, and this third cleavage when occur, it is the horizontal. The third cleavage is horizontal, and it form the 8-celled blastomere. It form 8-celled blastomere, and it occur again the 30 hours, or we can say that it is the 3rd days after the fertilization. Then after the 3rd cleavage, formation of 8 cell stage, and at the end of 4th day at the end of 4th day of the fertilization, the formation of 16 to 32 celled structure is formed, which is look like the mulberry, and that structure of 16 to 32 celled structure is known as the morula. It is the product of cleavage. The formation of morula takes place. It is mulberry structure, means it is the ball like structure, and again the size of the blastomere is decreases at the morula stage, and it occur at the 4th day after, means the end of the 4th day after fertilization, and the formation of morula takes place. Again in the morula, the zona pellucida is retained present. The zona pellucida is retained present. That's why the size is equal to the zygote. Means the size of morula is equals to the size of the zygote. Only the division of blastomere takes place and it forms the morula. After the formation of morula, it enter into the uterus. Means at the end of morula, at the end of cleavage, the morula is pushed towards the uterus through the isthmus. Means at the end of the morula stage, the morula reaches to the isthmus and it gains entry into the uterus, and this process is carried out by the end of 4th day. Okay, then morula, the second stage of the embryonic development is the blastulation. Means the formation of fluid filled structure, the formation of hollow fluid filled structure from the morula. From the morula is known as the blastula. And the process known as the blastulation, and the blastula is also known as the blastocyst. Again, the blastula or the blastocyst, it presents in the uterine cavity, and it absorbs the uterine milk, or we can say that the uterine fluid. The structure of the blastula is hollow fluid filled structure, and the formation of two-celled structure. The formation of blastula takes place, and the blastula contain two types of cell. The outermost thin cells and the inner cell of the mass is larger. We can say that it is larger inner cell mass, and it is covered by a small size cell that is known as the tropoblast. And the inner cell mass is attached to the tropoblast by one end, and the remaining cavity is known as the blastocyst cavity. The formation of blastula is from the 4th day to the 7th day after fertilization. The formation of blastula or blastocyst cavity is start at the 4th day, and the complete formation of blastula is end at the 7th day, in which the two types of cell is formed. In the hollow spherical cavity, there is the inner cell mass which is larger in size, and the outermost periphery cells, that is the tropoblast, and the inner cell mass is known as the embryoblast. It is known as the embryoblast. This inner cell mass is known as the embryoblast, and the outermost covering of the flattered radially cell, is known as the tropoblast. The development of the embryo in further development, this embryo develop from the embryoblast, from that the inner cell mass, and the embryo is proper developed from the embryoblast. Again, the blastula show polarity. The blastula show polarity. At the end, the embryoblast is present. At the end, the embryoblast is present, is known as the animal pole. Is known as the animal pole, or we can say that the embryonal pole. It is known as the embryonal pole, and opposite to the embryonal pole there is unembryonal pole. Okay, so at the 7th day it complete formation of the blastocyst, in which inner cell mass, that is the embryoblast, and it is covered by the radially cell which is known as the tropoblast, and these late blastocyst is formed at the end of 7th day, and it is ready for the implantation. This is ready for the implantation, and after the implantation it is ready for the gastrulation. It is ready for the gastrulation. Again, the zona pellucida layer is ruptured during the formation of blastocyst. At the early blastula stage it is present zona pellucida. The function of that zona pellucida is prevent the implantation of the embryo an abnormal site. It prevents the implantation of the embryo at abnormal site, and after the formation of blastocyst, which is ready for the implantation, and it implant in the uterus or it implant in the endometrium, and lastly the zona pellucida layer is ruptured and the blastocyst is ready to the implantation. Okay, in the implantation process, the implantation is also known as nidation. The implantation is also known as the nidation, in which the blastocele cavity of the blastula is present, and the inner cell mass or the embryoblast is attached to the endometrium layer. And this process, the process of the attachment of embryoblast in the endometrium cell, is start at the 7th day to the 10th day. The process of implantation is carried out 7th day to the 10th day, in which the blastocele is attached to the endometrium and it's embedded in the endometrium, and the blastocele cavity is remaining fluid filled, and the embryoblast form a chorionic villi, or that the embryoblast cell is formed the villi by which it is attached to the endometrium. At the cell or at the end of it is attached to the endometrium. That cell is known as the robber cell or cell of robber. It is known as robber cell. From the knob, it is attached to the endometrium and embedded in the endometrium, and the formation of villi or villi takes place, and the remaining blastocele cavity consists the embryoblastic cell as well as the tropoblastic cell. And this process of the nidation, or we can say that the implantation, is end at the 10th day after the fertilization, and it forms two layer. The implantation process is occur, or the implantation of the blastocyst is occur at the fundus of uterus. Means at the fundus of uterus, the blastocyst is embedded in the endometrium layer of the uterus, and it form lastly on the 10th day it form the two types of the cell. Outermost cell, which is known as syncytiotropoblast, The outer layer is syncytiotropoblast and the inner layer of the cell is known as cytotropoblast, and the inner layer of the cell is known as the cytotropoblast. And after the implantation or in the implantation, the embryo attached from the point, that point is known as the animal pole or embryonic pole, and opposite to the embryonic pole there is abembryonic pole, or we can say that the abnormal pole. Okay, so here first in the embryonic development, first one the cleavage and formation of blastula. Continue to the cleavage and formation of blastula, it formed the morula. It formed the morula has 16 to 32 cells. After the morula is divide and it form a hollow spherical cavity, hollow fluid filled structure, that is known as the blastula. Blastula is means the process of blastula, formation of blastula is occurred at the 4th to the 7th day, and after the 7th day the late blastocyst, we can say that the blastocyst is formed of 2 cells, and this 2 cell blastocyst is ready for the implantation, and at the implantation the blastocele cavity or blastocele is attached to the endometrium wall at the point of fundus, means is attached to the fundus at the point of endometrium wall, and it forms the villi. And after the implantation is ready for the formation of gastrula, and after the formation of gastrula, this process means from the blastocyst cavity to the formation of gastrula is known as gastrulation. Thank you.